Hello, in this video we will discuss about the Mendal law of inheritance. So, in this way, let's begin to understand first the Gregor Mendel. So, the Gregor Mendel was the scientist and priest, and in this way, he took the Piscium sativum plant, the Piscium sativum pea plant. Why he used the experimental plant Piscium sativum? Because it's normally self fertilization, so it can cross fertilization by itself, by their own aid, while on the other hand it can also cross fertilization by the help of humans. So that is why it can be changed the phenotype and genotype due to the cross fertilization by the help of human. So that is why it can be used as an experimental plant for detection of the inheritance of the plant. And on the other hand, traits can be phenotype of plants: round, wrinkled, yellow, green, and tall and small. So there are many phenotypes due to the genotypes, but there more than one traits basically. So that is why we can understand. So let's begin to understand. First is the cell, because Mendel didn't know about the cell structure inside of the genetic material as a chromosome. Here is the nucleus, and this is the cell wall. This is the chromosome. So chromosome are the genetic material are present within the nucleus and cytoplasm and vacuole. So anyhow, here is the nucleus which that contain chromosome, and this chromosome contain sister chromatids, which that one chromosome this is in the form of diploids come from the mother and father, for example. So in this way, here is the red and green gene producing, and this is the centromere, and this is sister chromatids, basically allele to each other, companion to each other. One can be recessive and one can be dominant, heterozygous or homozygous, homologous and heterologous, and homozygous and heterozygous. In this way, let's begin. This is the locus, contain many gene is known as loci. Locus means single. So the green and yellow and tall and small show in the small t and capital T, and large G is green and Y is the yellow color, while the round and wrinkle on the basis of the letter, capital letter and small letter. So these chromosome contain a nucleosome. This nucleosome contain histone, protein, and DNA. This DNA contain a gene. For example, here is the gene 1 to 14. So these different genes contain a thymine, cytosine, guanine, and adenine, the nitrogenous base, which that is produce the gene. So after this DNA transcription to produce messenger RNA. After this messenger RNA go into the cytoplasm to produce the protein for production of color due to substance synthesis and shape due to different protein combination. So this all thing is due to the genetic material which that show the phenotype from the genotype. So yellow or red basically are round, yellow and round, as well as green and wrinkle basically. So the laws of Mendel was two: law of independent assortment and law of segregation. Let's begin to understand the law of independent assortment first. So this is I am drawing the chromosome which that contain a yellow and green color, contain a homozygous condition, heterozygous condition, sorry. So heterozygous means the two different alleles are present in the same locus, in the same alleles in the different locus. So in this way, here is the round and wrinkle gene are present. So the round is dominant and yellow is dominant on the Y, and round is dominant on the wrinkle. So if the Y gene are present and R gene, so the yellow and round will produce not wrinkle and green. So this is the gamete formation. After during the gamete formation to produce haploid, this haploid will be independently segregate. After this segregate, the chromosome will again during the fertilization will unite to produce different traits. For example, Y and small y and Y and small y. round and round. This is the homozygous condition and this is the heterozygous condition. So this round and yellow, this and round and green, due to the homozygous and heterozygous condition. If the wrinkle is recessive, so the both genes should be wrinkle for the dominancy for wrinkle production. While the round gene are present, so the wrinkle will be recessive. It means the round is dominant on wrinkle. So let's begin to understand the law of segregation. So that was the independent assortment, but this is the segregation is same, but you can see this is the segregation and the round, wrinkle and round gene will be separate in the gamete formation, for example, sperm and egg. So after again fertilization to produce new combination through RR, R small r, and R small r and small r small r. It means the one is wrinkle while the two also wrinkle round but it is heterozygous. And homozygous is double R. It means both are dominant and the homozygous dominant and heterozygous dominant round, while the homozygous recessive allele will produce wrinkle structure, wrinkle uh, bean or wrinkle pea, wrinkle seed. So this round and wrinkle. It means the 3:1 is the phenotype, while the genotype is 2:1:1. So remember these things. Let's begin to understand the definition. So the after law of independent assortment is the after segregation allele will be independent during gamete, but after fertilization it will depend on dominance. So it will depend, for example, the bigger large bigger capital R is the round and small r when combined together, so the capital R is the round will be dominant on wrinkle. So here is the definition of the another law of segregation. So the alleles are segregated during gamete formation and randomly unite again at fertilization to show new dominance alleles. So in this way the new allele will be shown. So basically both are same, but the more than one trait involved in the independent assortment. So here is, let's begin to understand the cross. So the homozygous dominant and homozygous recessive allele will fertilize to after gamete formation to produce parental generation to the F1 generation, that is the heterozygous condition round. So its round is basically is the 100% round, but genotype of this contain also the wrinkle gene. So in this way here you can see the Punnett scale rule which that apply to show the phenotype again the F2 generation through a gamete formation R and small r will combine together to you should understand this function. So the phenotypic ratio 3:1 while the genotypic ratio 1:2:1. So the Punnett scale rule to show the self cross of F1 generation, remember. So let's begin to understand the dihybrid and dihybrid cross. So the dihybrid means that more than two or more than one phenotypes which that is produced from genotype. So for example, this is the round as well as yellow. So it means the bigger capital R containing both homozygous condition, while the small capital Y show the yellow color as a phenotype from the genotype homozygous. While this is a homozygous recessive contain wrinkled and green due to the small r both and small y. So in this way you can see this is the round and yellow. So remember this is the round and yellow which that is here. Let's begin to understand the Punnett scale rule. In this way this was the F1 generation which that is produced all is the yellow, but it's contain a gene which that is further F2 generation will show the genotype as a phenotype from the genotype. So let's begin to understand F1 cross. So in this way, F1 here is the possibilities of the gamete can be capital R and capital Y and capital R and small r and large Y and capital Y and small r and small y. We can understand as well. And horizontally and vertical show equally the gamete. So in this way, the first cross will show the capital R and capital Y homozygous condition to show yellow color of the seed and round because capital R. And here is the capital R both homozygous and the heterozygous condition which contain capital Y, so it means the yellow. And whole this process you can understand after this cross. So in this way the Punnett scale rule can understand there are different varieties and their phenotypes will be different due to the genotype. So this will be segregation, and this color will produce again also produce the green color due to the again dominancy, and again the law of segregation will apply these things, law of independent assortment. And in this way you can understand this is the basically the round and yellow 9 and round and green 3, wrinkled and yellow 3, and wrinkled and green 1. It means the 9 ratio 3 ratio 3 ratio 1, which that is the F2 generation will produce, and in this way the round and yellow will again dominate. So the gene pool contain a same gene, but the phenotype will be different on the basis of dominant and recessive due to the gene influence, the dominant influence. So thanks for watching. Bye.